Hello brainers, my name is Jason. Welcome to the Big Brain Busters. So in this video we are going to look at the functional unit of the muscle tissue, and as usual at the end of this video we have put up some practice questions that I would like you to attempt and see whether you've actually understood. If time allows us, we are going to attempt those questions together. With that being said, let's jump into it. Okay, so now let's kick it off by knowing what a muscle is. So this is simply a soft tissue that is made up of many stretchy fibers whose function is to provide movement through the mechanism of contractibility. So we have three main types of muscles in our body, and these are the skeletal muscles, the smooth muscles, and the cardiac muscles, of which the skeletal muscles are the muscles that we use to voluntarily make movement. The smooth muscles are those that provide contraction to our internal viscera or our internal organs. Then the cardiac muscles are those that provide contractility to the heart. They are also known as the heart muscles. Okay, so now let's talk about the organization of a muscle tissue. So starting from the gross aspect to the histological aspect of the muscle tissue, we are going to use the skeletal muscle in this video as our reference to learn the organization of the muscle tissue, because the skeletal muscle is the one that shows better imaging in terms of studying, so it shows the structures more clearly. So as you can see here, starting from the bone where the muscle attaches via a tendon. Okay, a tendon is a connective tissue. So as you can see there, this is a tendon attaching to the bone. Then this tendon is a continuation of a connective tissue covering known as the epimysium. The epimysium covers this entire muscle. This entire muscle here, it is covered by the epimysium. And this entire muscle is simply a group of muscle fibers known as fascicles. So these structures in here are known as fascicles. Okay, and these fascicles are actually covered by a structure or a connective tissue covering known as the perimysium. Okay, then from there, this fascicle is a group of other muscle fibers inside here. Okay, these round structures here. So this is the fascicle, and it is a group of the round structures that I was drawing there, and these are known as the muscle fibers. Okay, and these muscle fibers are covered by a structure or a connective tissue layer known as an endomysium. So this diagram is a continuation of this guy. Okay, it's a continuation, so don't get confused. So we are on the fascicle. Then a fascicle is a group of muscle fibers. These are the muscle fibers inside the round structures there, which are projecting outwards. So this is so here we have it. We have the muscle fiber projecting outward like that, and it is covered by a connective tissue known as the endomysium. Okay, then this muscle fiber here is a group of myofilaments known as the myofibril there. Okay, so these are the myofilaments. It is composed of myofilaments such as actin and myosin. Okay, then the epimysium here outside, it covers other neurovasculature such as arteries and veins, and these arteries and veins, they pierce through the fascicle to go and provide blood supply and innervation to the muscle fiber. So this is the the neurovasculature that I was talking about here. So you can see we have got some arteries in the nerves there. Okay, so now this is just simply another representation of organization of a muscle tissue. So at first we say that we have a muscle tissue that is connecting to the periosteum of the bone, and this muscle tissue is actually covered by the epimysium. Then from a muscle tissue we say that it is a group of muscle fibers known as what? A fascicle, and the fascicle is covered by a layer of connective tissue known as the perimysium. Then this fascicle is actually a group of muscle fibers. Okay, and this muscle fiber is actually known as what? Is actually covered by the endomysium. Then from a muscle fiber, the muscle fiber is made up of other structures known as the what? The myofibrils, which are the myofilaments. Okay, and the myofilament or the myofibrils are made up of myofilaments such as myosin and actin. So in that order, if you want, you can make a mnemonic. I'm not that good with making mnemonics, but yeah, all I know is that there are so many M's in this organization. Anyways, let's continue. So now let's look at the functional unit of a muscle cell. So the functional unit of a muscle cell is actually known as a sarcomere, all right, which is a basic unit of a striated muscle. So why are we saying striated muscle? Because it is limited. Remember that we said we have three types of muscles in our body. So the sarcomere is only found in the cardiac and skeletal muscles. All right. So this is where a sarcomere is found, because the sarcomere forms striations. So that's why we say it is a basic unit of a striated muscle. So take note that a smooth muscle is not striated. That's why it's called a smooth muscle, because it does not contain a sarcomere which provides striations. Okay. So note that a sarcomere is not found in smooth muscles. Why? This is because there is no regular arrangement of actin and myosin filaments. Then instead, the actin filaments are actually arranged in an oblique direction in the cytoplasm. Okay, now take note that the cytoplasm of a muscle tissue is known as a sarcoplasm. Then the cell membrane of a muscle tissue is known as a sarcolemma, right? So now let's look at these myofilaments. So these myofilaments, we have the thin filament known as the actin filament or the actin protein, right? These are the structures. And then the thick filament is known as the myosin. Okay, so the actin is actually composed of actin protein, right? Then this is what you're able to see here, and this actin protein is actually made up of repeating units of G actin and F actin, so as you can see, and it is a spiral. It is composed of two polypeptide chains which are wound around each other, which are spiral-like, and it is also composed of two regulatory proteins known as tropomyosin and troponin. So the tropomyosin is this green structure that you can see winding around the two polypeptide chains of actin. And this tropomyosin, it covers the binding sites where myosin can bind. So as you can see, there are like these small dots around, and that is where tropomyosin is covering. So it is covering there whenever there is no binding or whenever there is no contraction. So as you can see, this is the myosin binding site, and tropomyosin is covering there, right? Then we have troponin, another regulatory protein. It is composed of three subunits. So it is composed of troponin C, troponin I, and troponin T. Okay, so these are just some notes of what we're talking about. So the actin filaments have binding sites for myosin cross bridges. Yeah, we've already mentioned that. Then the tropomyosin protein spirals around actin helix, covering the active site on actin. So here, this is what we're just talking about. So the tropomyosin covers the binding sites for myosin binding site. Then the troponin is composed of three subunits, which are troponin T, troponin I, and troponin C. And the troponin C, that is where the calcium binds. So how I remember, because C for calcium, right? Then nebulin, an accessory protein, which is one of the largest proteins in the body, it joins the filament, the actin filament, to the Z disc. You will understand that in the later section of this video. Then this entire complex of actin is known as the troponin myosin complex. So now let's talk about the thick filament. Remember, the thin filament is the actin filament. Now we talk about the thick filament, which is the myosin filament. So this is the thick filament. It's made up of So this is a myosin molecule, which is made up of also two polypeptide chains which are wound together, but then it has two things, tapering at the end. So these are the myosin heads, and these myosin heads contain actin binding sites and ATP binding sites. Okay, so these myosin filaments, they are actually connected to the Z disc by the largest protein in the body, known as titin. You will see that when we look at the sarcomere in detail. So now, what are some of the muscle proteins that are found there? So we have the contractile proteins, which are the myofilaments such as actin and myosin. Then we have the regulatory proteins, which are tropomyosin and troponin, found on actin. Then we have the structural proteins that provide, integrity to the myofilaments, and these are titin and nebulin. We said titin is the largest protein or accessory protein in the body that attaches the myosin filaments to the sarcomere, and nebulin attaches the actin filaments to the sarcomere, which is the Z disc. They both attach these myofilaments to the Z disc. Okay, so now this is the general arrangement of a sarcomere. Alright, as you can see, this is a myofibril. So this entire thing is a myofibril, and then it is made up of these striations, and this is a sarcomere here. Alright, okay. So now this is the microscopic anatomy of the skeletal muscle. So as you can see there, we have the the striations and everything. So this is just showing you the how under a microscope this will look like. Okay, or when you zoom in on a myofibril, then it is not different. It is not much different with a cardiac muscle, because the cardiac muscle is also made up of the sarcomere, just that it is also composed of intercalated discs, and these intercalated discs, you find gap junctions, and the function of gap junctions is just simply for communication between different between different cardiac muscle fibers. Okay, so now let's look at the components of a sarcomere. So the sarcomere is made up of different components. So as we said, the main ones are simply the thin or actin filaments and the thick myosin filaments. So these are the myosin filaments, these are the actin filaments. We've already talked about them in detail, how they are composed. Okay, then this is a sarcomere. A sarcomere is simply running from one Z disc to the next Z disc. This entire thing here is known as a sarcomere, whatever is composed here. Okay, after the Z disc is another sarcomere. All right, and this diagram down here shows a sarcomere and how it looks like under a microscope. So the sarcomere is composed of a thick or dark band and a light band. Here, as you can see, it's like showing white, and here it's showing more darker. So the dark part here of the sarcomere is known as the A band, running from this point to that point. Okay, this is the A band. Why is it called the A band? Because it is composed of both the thick and thin filaments. That is why it appears dark. So as you can see there, so this is why it's appearing dark. Then in this A band, you find a zone known as the H zone. So the zone here, which is from here to there, from here to here, this is the H zone. Why is it called the H zone? Because it is only composed of the thick filaments. As you can see, these thin filaments are not found here in the H zone. They are only thick filaments, right? So that's why it's called the H zone. And in the H zone, there is an M line which bisects the H zone. This M line here is composed of a protein known as myomycin, this M line here. Okay, so as you can see, the H zone appears lighter because there is no thin filaments, only thick filament is there. Then what else? We have also this light part here. Okay, so this light part, which only contains thin filaments, is known as the I band. So this is the I band here. It's light stained because it only contains thin filaments. Okay, and in this I band there is a Z disc which bisects this I band. Okay, this is the Z disc, and this Z disc is where the myofilaments attach to. Remember in the previous slide we are talking about the titin and nebulin proteins. So titin proteins are the ones that attach the thick filaments. So this green thing here, this green protein attaching to the Z disc is known as titin, and it is the largest accessory protein in the body. Then we have nebulin. Nebulin attaches, it's just there. I think they haven't labeled it in this diagram. It attaches the thin filaments to the Z disc. Okay, then another structure that we can talk about, I think we've talked about everything here. All right, cool. So we move on. So this is a sarcomere in a normal state, and this is a sarcomere in a contracted state. So this leads us to the next thing that we talk about, in short, the events that occur during contraction of a muscle. So there are certain things that occur in the sarcomere, and as you can see here, the two differences. So in general, whenever a muscle fiber contracts, the sarcomere shortens. Okay, the sarcomere shortens. And remember, we said that the sarcomere is simply a distance between two Z discs. So from this distance to this distance, this entire distance here shortens. But take note that the myofilaments do not shorten, they remain the same. Okay, so as you can see here, from this distance to this distance, it has been shortened. Then another change that you can take note of is the H zone. So here in between, there is the H zone. The H zone has been reduced. So why has it been reduced? Because the actin filaments have been drawn together and now they are overlapping, so meaning that we have no H zone here. Okay, then another change that you can take note of is that the Z discs have been brought closer together. That's why it is making the entire sarcomere to shorten. Then another thing is that the I band will be shortened as well. So as you can see here, the I band has been shortened, but the M line still remains in the middle and it still remains intact. Its length is not changed. Okay, same as the Z line as well, its length is not shortened, but the two Z lines, they are actually what? They are actually brought together. Then the A band, the distance of the A band is not changed during contraction. It remains the same. What changes is that the A band of this sarcomere and the A band of the next sarcomere, their distance is actually brought together. Okay, so these are the changes that occur in contraction of the muscle fiber in a sarcomere. So this is just simply a summary of a relaxed and contracted muscle fiber, which shows what happens to the sarcomere. So the A band stays the same, the I band gets smaller, the H zone gets smaller, the sarcomere shortens, but take note that the filaments do not change in length. Okay, so now these are just some notes that I've added. You can pause the video and try to write down these notes. They'll be of help, especially when you want to study alone, in your books. Same as this. Now let's look at some clinical correlations towards, the functional unit of a muscle fiber. So you have a condition known as myasthenia gravis. So myasthenia gravis is simply an autoimmune disease. So an autoimmune disease meaning that it is a disease caused by our own body. So what happens is that our own body will produce antibodies. So these antibodies will go and destroy the acetylcholine receptors where acetylcholine is supposed to bind. And then when that happens, there won't be contraction of the muscle occurring and thus leading to muscle weakness and fatigue. So that is what happens in myasthenia gravis in simple terms. Then Duchenne's muscular dystrophy is another, this one is actually a congenital kind of disease in muscles whereby an individual lacks a protein known as dystrophin. So the dystrophin protein is actually important in providing integrity of the sarcolemma. Remember that we say the sarcolemma is simply the cell membrane um, of the muscle tissue. So that sarcolemma during contraction, it is the dystrophin protein that provides that integrity during contraction so that it can withstand the stress. But then in people with DMD, these people actually do not have that protein. Hence, it also causes muscle weakness during contraction and fatigue. Okay. So these are some of the practice questions that I have put about. So we've actually covered about 20 minutes of this video, and I wouldn't want this video to be that long, but you can try to attempt these questions, and if you have any question or anything, you can drop it in the comment box. And I would actually love if you were to drop your answers in the comment box, then I will definitely reply to tell whether they're actually right or wrong. These questions are all true or false kind of questions, and I think everything that is in this question is covered in the video. Only maybe one or two things that if you won't understand, you can drop it in the comment box. Alright, so this is where we end our video. Please don't forget to like, share, and subscribe to our YouTube channel, and also check out our other videos. With that being said, let's end here. Shalom, shalom.